So now I would like to discuss again what is the blood coagulations or blood clotting mechanisms. So what is the blood coagulation? When blood is shed, it loses its fluidity in a few minutes, okay, and sets into a semi-solid jelly. This phenomenon is called the coagulation or clotting of the blood. On further keeping, the clots retract to a small volumes and passes out as a clear straw color fluid called the serums. It is to be noted that coagulation is the property of the blood plasma and the platelets alone. The red and the white cells do not take part in it. Okay? They only become caught up in the meshes of the clots and are thereby removed. So blood coagulation mechanism is mainly deals with the blood plasma and the blood platelets. Okay? Red blood cells and white blood cells nothing to do. Okay? Next. Whenever blood vessel is ruptured, prevention of the blood loss is achieved by several different mechanisms such as vascular spasms, formation of the platelet plug, eventual growth of fibrous tissue into the blood clot. Over 50 important substances that affect blood coagulations have been found in the blood and the tissues. Some promoting coagulations called the procoagulant and other inhibiting coagulation called the anticoagulant. The normal bleeding time is 2 to 5 minutes. So I have given one diagram. The diagram you can understand what is a blood, that is coagulations. Okay. So in these diagrams, I have mentioned that injured tissue and broken platelets, first it will be released at the spot of injured blood vessels. As a result, there is a liberation of thrombin protein which is popularly known as thromboplastins. Now, this thromboplastins with the help of the calcium divalent ions, factor 7 and 10, active factor 7 and 10, it converts plasma protein which is the prothrombin into active thrombins. Okay, prothrombin is the inactive proteins and thrombin is the active proteins. Okay. Now, when there is a thromboplastin from the injured tissue, there is a release of the thromboplastin. This thromboplastin increases the calcium ion, factor 7 and factor 10. It converts prothrombin into thrombins in the plasma. Okay. Next, another protein which is on the fibrinogens. This is also inactive. This fibrinogen converted into fibrin active protein again with the help of the thrombin proteins. Okay. Now, this fibrin is a monomers. Okay. So many fibrin, it will plug the rim of the blood vessels. Okay, it will stitch the blood vessels, which is popularly known as the polymers. Fibrin monomer it will convert into polymer, and ultimately clotting takes place. Okay, and WBC or RBC was stuck in that meshes. So this is actually the figure which that is blood clotting mechanisms. Okay, so let us discuss the blood mechanisms in text. Okay. When blood is shed, the blood platelets disintegrates and liberate the thromboplastins. Okay, certain amount of the thromboplastin is also derived from the damaged tissue of the injured locality. Thromboplastin converts prothrombin into thrombin with the help of the calcium divalent ions. Thrombin interacts with the fibrinogen to form the fibrin monomers. Okay. Now, fibrin monomer from the fibrin polymer which flags the rim of the blood vessels. The mechanism of the coagulation involves activation, adhesion, and aggregation of the platelets as well as depositions and maturation of the fibrins. Basically, blood clotting takes place in 3 steps. In the first step, the blood platelets and the blood plasma forms the tissue thromboplastins or prothrombin activators at the spot of the injury. Now, thromboplastin or prothrombin activators converts prothrombin into thrombins. In the next third stage, the thrombin converts fibrinogen into fibrin monomers, and fibrin monomer from the fibrin polymer, and it plugs the rim of the blood vessels. So this is the actual mechanism of the blood coagulations. So in my next slide, I have shown you 13 factors. According to International Committee of the Blood Coagulation, requires 13 factors which are as follows. Factor 1, which is popularly known as the fibrinogen. Factor 2, that is prothrombin. Factor 3, tissue thromboplastins. Factor 4, calcium ions, divalent ions. Factor 5, level factor or the proaxillary. Factor 6, axillarin. Factor 7, there is stable factor or a pro-convertin. Factor 8, there is the anti-hemolytic factors. Factor 9, plasma thromboplastin components or Christmas factor or platelets cofactor 2. And the factor 10, there is Stuart factor. Factor 11, plasma thromboplastin antecedents. Factor 12, Hogman factor or Stuart factor or clot promoting factor. And the factor 13, that is known as the Lucky-Laurent factor or fibrin stabilizing factor or fibrinase. So, according to International Committee of Blood Coagulation, these 13 factors are essential for blood coagulations. Okay. Now, these 13 factors, it acts indifferently. Some acts in the extrinsic way, some acts in the intrinsic way, some in a common pathway. Okay. Ultimately, it helps in the what? That is blood coagulations. So, in my next slides, I will show the basic mechanism of the blood coagulation. It can be studied on the following steps. Number 1. That is known the primary homeostasis involves the first two processes. Okay, there is a vasoconstriction and platelet plug. Vasoconstriction means the blood vessel it will constrict. That is the vasoconstrictions. Okay, vasoconstriction is the body's first response to injury the vascular wall. When injury occurs, vessel's wall constricts. Blood vessel wall constrict, causing reduced blood flow to the site of the injury. So, this is the first function, this is the vasoconstrictions. Second is the platelet plug. Platelet, what is a platelet plug? Platelets aggregates to the site of the injury. They stick together acting as a plug. Platelets also activate the process which causes the fibrin clot to form, okay, known as the secondary hemostasis. So, these two are popularly known as the primary hemostasis. Next, secondary hemostasis, it will be B, not A. First one is A, primary, and secondary will be the B. Okay. Now, platelets alone are not enough to secure the damage in the vessel walls. Okay, a clot must form at the site of the injury. The formation of the clot depends upon several substances called the clotting factors. Already I have mentioned 13 factors are there. These factors are designated from the Roman numerals from Roman 1 to 13s. Okay, 1 to 13s. These factors activate each other in what is known as the clotting cascades. Cascade of the reactions, one after one. The end result of this cascade is the fibrinogens. A soluble plasma protein is clipped into a fibrin and non-soluble plasma proteins. Okay. The fibrin monomer, it will form the fibrin polymer into a fibrin polymer which plugs the rim of the blood vessels. Okay. So in my next slides, I am going to show the clotting mechanisms. The clotting mechanisms or the clotting cascade reaction occurs through 3 separate pathways that interact the intrinsic, extrinsic and common pathway and result in the blood coagulations. Now, what is the extrinsic pathway? Extrinsic means outside the blood vessels, intrinsic means inside the blood vessels. Okay. The extrinsic pathway is achieved by the external trauma that causes blood to escape from the vascular system. This pathway is quicker than the intrinsic pathway. It involves the factor mainly 7s. Okay, factor 7 plays the main role in the extrinsic pathway. In the intrinsic pathway is activated by the trauma inside the vascular systems and is activated by platelets exposed to endothelium, chemicals or collagen. The pathway is slower than the extrinsic pathway but is very important. It involves factor 12, factor 11, factor 9 and factor 8. Okay. After the extrinsic and intrinsic pathway, there is another pathway in the common pathway. Both the pathway enters in a common pathway. That is a popularly known the common pathway. Both pathways meet and finish the pathway of the clot formation in what is called the common pathway. The common pathway, it involves the factor 1, 2, 5 and 10 and 12. And the other factors are calcium divalent and phospholipids. Okay. So, these all factors, okay, simultaneously they act ultimately in the extrinsic way, intrinsic way, common pathway. Ultimately, it helps in the blood coagulation of the blood clottings. So, I have shown a schematic diagram of intrinsic pathway, extrinsic pathway and a common pathway. Blue one is my left hand blue one, it is known the extrinsic pathway, intrinsic pathway, and that pink color that is known the extrinsic pathway, and this yellow color is known as common pathway. So ultimately it is whatever may be the pathway, ultimately all the three ultimately coincide together. Ultimately it leads to the what? Stable fibrin clot formation or a fibrin monomer formation, and this fibrin monomer ultimately it will give rise to a fibrin polymer, and this polymer ultimately what will happen, it will plug the rim of the blood vessel from the meshes and plug in the big blood vessels, and ultimately blood coagulation take place. Now if any factor is not working properly, then there will be hasten of the blood fluctuations. Okay, and there are some in some families it is a disease, the blood factor is not properly acting, ultimately blood coagulation is delayed. So you might have noticed that is it is the royal families of the that is Great Britain, blood coagulation is delayed. Okay, so in my next slide, again the same thing I have shown you regarding the intrinsic pathway and the extrinsic pathway. Say from the extrinsic pathway, tissue injury, it will give you a tissue factor. Ultimately 7 is inactive, it will give us to the 7 active form. Similarly here in intrinsic pathway 12, then 11, then 9, then 10, all it will convert into active form, and ultimately they will give rise to the prothrombin into thrombin, and this thrombin ultimately help in the fibrinogen into fibrin monomer, and when fibrin monomer formation takes place, fibrin ultimately gives rise to the fibrin polymer into factor 13 and vitamin K, and ultimately it leads to a fibrillation, that is clot formations. So this is the whole mechanism of the blood coagulations. Okay, so I think it is clear. With that I would like to conclude. So from the blood I have discussed structure and composition of the blood. Okay, detailed structure and composition. Then I have discussed regarding the blood groupings. Then today I am going to blood groupings 2, that is the ABO blood grouping and the RS blood groupings. And today now I am going to discuss, I have completed regarding the blood coagulations. So this chapter is, so far this blood is concerned, this chapter is over. Okay, so with that I would like to conclude. So if you face any problem, let me know.